Welcome to this explainer. Today we're diving into a 2026 Nature Microbiology study that honestly sounds a bit like science fiction. We are going to explore how researchers have literally uncovered life-saving antibiotics hidden right inside the very proteins we typically associate with fatal, incurable brain diseases. It's an amazing story of deep learning, computational biology, and a massive biological plot twist. Let's see how they actually pulled this off. So, let's kick things off with a totally counterintuitive question that sits right at the heart of this research. Can a deadly protein actually save lives? For decades we've viewed amyloid genetic sequences. Those are the sticky fragments that cause proteins to misfold and clump together into plaques as strictly the bad guys. But in biology, things are rarely so black and white. Researchers started wondering, could these aggregation-prone proteins actually be encoding cryptic, host-protective fragments within their primary sequences? It's a wild premise, and it completely flips our conventional understanding right on its head.
It's just a massive biological juxtaposition. On one hand, you've got classical prions. These are the terrifying agents behind conditions like mad cow disease, notorious for corrupting healthy proteins and destroying the brain. But on the other hand, we have antimicrobial peptides, or AMPs. These are central effectors of our host defense, basically the good guys that actively kill drug-resistant pathogens. Historically, these two camps have been viewed as completely separate worlds, but recent murmurs in the scientific community hinted that some amyloid-associated proteins might actually display antimicrobial activities. The catch was nobody had systematically checked to see if these hidden or encrypted peptides were broadly embedded across all prion and prion-like proteins. Well, until now.
To find out if these known villains were secretly harboring a host-protective alter ego, researchers needed some serious computational muscle. They deployed a deep learning framework called Apex 1.1. First, they curated nearly 3,000 UniProt entries that were annotated as prion or prion-related, and then computationally chopped them up into all unique 8 to 50 residue fragments. That yielded a staggering 19.3 million fragments. They were essentially mining a vast, totally unexplored biological database, searching for a very specific, microscopic needle in a haystack.
They effectively built a massive computational funnel to get from this giant sea of data down to tangible, life-saving molecules. Starting with those 19.3 million fragments, they ran them through the Apex AI to predict their efficiency against an 11-pathogen panel. The AI narrowed it down to 1,179 highly promising candidates. From there, to see if this actually worked in the real world, the team synthesized 75 of the most diverse peptides, and the results? Absolutely stunning. In lab tests, 59 of those 75 synthesized candidates actively inhibited clinically relevant, often multi-drug-resistant bacterial pathogens. For computational biology, that is an incredibly high hit rate.
And that brings us to the big reveal. The researchers coined a brand new term for these encrypted defense fragments, Prionins. Just take a second to appreciate what this actually means. These aren't entirely new proteins from scratch. They are functional bacteria-killing sequences encrypted directly inside the primary sequence of prion and prion-like proteins. It's literally like finding a hidden code inside a computer virus that actually runs as antivirus software.
Now, you might be thinking this is just some weird anomaly confined to one specific animal, but no. These newly discovered prionins are broadly distributed across the entire tree of life. The candidates the AI found came from 1,068 different organisms. We're talking about fungi, multicellular animals, and single-celled organisms. They derive from canonical prion proteins, prion-like glutamine and asparaginine-rich proteins, and even prion inhibition proteins. This tells us that this encrypted antimicrobial potential is an evolutionary strategy embedded widely across the entire prion-associated sequence space.
But here's a question. Are these prions just carbon copies of the antimicrobial peptides we already know about? Not quite. When researchers computationally mapped these out, they realized prions occupy a distinctly new biological neighborhood. They're longer, they carry more of a positive charge, and they repel water differently than classic known AMPs. They just don't follow the standard structural blueprints we're used to seeing. What that means in plain English is that these prion-derived fragments are a genuinely new flavor of antibiotic.
So how exactly do these prionins destroy superbugs? Well, the easiest way to visualize their mechanism of action is basically popping a bacterial balloon. Classical antimicrobial peptides often work by disrupting the bacterial membrane, and the researchers really wanted to see if prionins follow a similar playbook. Spoiler alert, they absolutely do. And they do it with ruthless efficiency, from the outside in.
The actual molecular mechanics of that popping process are super fascinating. Lab tests prove that these peptides are mostly unstructured while just floating around in water. But the second they touch a target bacteria, boom, they fold up, activate, and act like microscopic drills. They permeabilize the outer wall and completely collapse the bacteria's internal energy potential. In many cases, these encrypted fragments destroy the bacteria's structural integrity just as effectively as standard, highly potent reference antibiotics. Now, if you follow biological research, you already know what the next logical hurdle is. It's one thing to zap bacteria in a carefully controlled petri dish, right? But does it actually work in a living, breathing subject? A massive barrier in antibiotic development is host toxicity, because many things that kill bacteria also destroy our own cells. So before even thinking about animal models, the researchers had to heavily screen these candidates for safety. Out of that synthesized batch of 75, a select few proved to be highly selective, meaning they completely annihilated the bacteria, but safely ignored the host cells. The team zeroed in on 2 lead candidates for testing: Prionin 7, which actually comes from a type of fungus, and Prionin 38, derived from a microscopic roundworm. When tested against human red blood cells and human embryonic kidney cells, both of these candidates showed 0 detectable toxicity, even at the highest tested concentrations. They were officially safe enough to advance to a live mouse model.
And wow, the live trial was an incredible success. The researchers used a mouse skin abscess model infected with Acinetobacter baumannii, which is a notoriously difficult-to-treat superbug. While the untreated control group suffered a massive bacterial burden by day 2, the mice given just a single topical dose of either Prionin 7 or Prionin 38 showed a drastic reduction in bacterial load. Their efficacy closely tracked with polymyxin B, a powerful standard of care antibiotic used as a baseline. The deep learning model's predictions held up flawlessly in the real world.
Plus, and this is absolutely critical for any future drug, this safety profile held up perfectly in the animal model. The mice treated with these prion-derived peptides stayed entirely healthy. Researchers closely monitored them and noted absolutely 0 treatment-associated weight loss or any visible signs of distress. These encrypted sequences were acting purely as precision anti-infective agents, leaving the mammalian host completely unharmed. Which brings us to a really profound conclusion directly from the authors of the study. They state, "Our findings identify prion and prion-like proteins as an unexpectedly rich reservoir of encrypted AMPs. This extends a wildly exciting new concept in biology. The sequence features underlying aggregation-prone, disease-associated proteins can actually coexist with, or even give rise to, defensive antimicrobial fragments. It proves that unconventional protein classes can be a massive, completely untapped source space for antibiotic discovery, right when we desperately need new ones." As we wrap up this explainer, I want to leave you with a pretty provocative thought. What other life-saving cures are just hiding inside the very things that cause us harm? If deep learning can look at a devastating prion protein and extract a pristine, functional antibiotic from its code, what else is lying dormant within our own biology? The potential for AI to unlock nature's encrypted medicine cabinet is truly just beginning. Thanks so much for joining me to explore this fascinating data, and I'll see you in the next explainer.